Now here we'll talk about two different proteins known as the major histocompatibility complex proteins, or the MHCs. And these are huge proteins that are found on the cell membrane of basically all of our cells. There are some exceptions that we'll talk about in a minute. And that huge protein is known as the major histocompatibility complex. It is coded, these proteins are coded by a gene found on chromosome 6, and we all have an identical, or we all have unique MHCs. None of us are identical unless you have an identical twin. This is how our immune system knows that this is a self cell or my cell and I'm not supposed to attack it, but it has the ability to recognize an MHC of a foreign cell and attack that one. And that's why when we perform an organ transplant, you want to get cells that are as close as possible to your own MHCs. So if there's no identical twin, well, maybe a sibling or a parent and so on. So basically your MHCs are the genetic markers of your biological self. And there are two different kinds of MHCs. There's MHC1 and MHC2. And you can see here, that's the cell membrane, and you can see how that protein is a very complex protein. You don't need to worry about what it looks like. I just have this up to show you the complex structure of that protein. And MHC1s are found on all of our cells with the exception of our red blood cells. So our RBCs do not have MHC1s. MHC2 cells, those are the cells, these are the protein complexes that are more related to the immune system. Those are found on the macrophages, B lymphocytes, and dendritic cells. Dendritic cells are the macrophages found in the CNS. Now, let us see how these MHCs work. Remember, the T helper cell was the cell that had to activate the B lymphocyte and the cytotoxic lymphocyte. The only problem is that the helper T cell needs another cell to present the antigen to it. So you see here this antigen presenting cell presenting the antigen to the helper T cell. Now this helper T cell will start making different cytokines, activating the B lymphocytes and the cytotoxic T cell. These are some restrictions on how these MHCs work. So for example, B lymphocytes, they are not going to interact with MHC proteins. They are actually going to interact with antigens using antibodies. Helper T cells can bind only to the class 2 MHC. So helper T cells are going to interact with the class 2 MHCs that are already found on your B lymphocytes, macrophages, and dendritic cells. Helper T cells has a nickname, and that nickname is CD4. CD stands for Cluster of Differentiation 4, and we're not going to really get into the details of what CD4 means. But you want to know that helper T cells are also known as CD4 cells, and those bind to class 2 MHCs. While your cytotoxic T cells, those are known as CD8. These can only bind to class 1 MHC. And remember, class 1 MHC is found on any cell with the exception of RBCs. Natural killer cells do not need to interact with MHCs in order to become activated. And we are going to talk a little bit more in detail on how these MHCs work and how they activate the different cells. Just a little mnemonic here to remember which one activates or interacts with which. The CD4 activates with class 2 and the CD8 interacts with class 1. So when you multiply them, we always get an 8. Okay, so CD4 times 2 is 8. CD8 times class 1 is 8. So we will take a look how, remember that helper T cell needs an antigen presenting cell to present the antigen to it. So I'm just going to take you back some slides where we encounter and recognize the cell, recognize the antigen. B cells are going to recognize antigens because they are marked with antibodies, while the cytotoxic T cells are going to recognize pathogens presented to them on the the MHC ones, while the helper T cells, they really don't have the ability to recognize a cell on their own. These cells have to be presented to them on the MHC2. And because MHC2 is only found on macrophages, B and dendritic cells, those are the cells that are going to act as antigen presenting cells. So you can see here a macrophage that is going to engulf the antigen. It'll break it down and then take a little part of the antigen and present it on its class 2 MHC on the surface membrane of the macrophage. Now the T helper cell has a receptor that is able to identify the class 2 MHC. Had this class 2 MHC, if it was empty, then there would be no problem. But because it's not, that is going to lead to the activation of that helper T cell. Here is your B lymphocyte. And you can see here that in order to activate the B lymphocyte, there is no interaction with with any MHCs on the antigen. Actually, the antigen is flagged by an antibody. The B lymphocyte engulfs it, it digests it, and again presents a little portion of it on its MHC2, on its class 2 MHC. And then that class 2 MHC, again, will be identified by the helper T cell and activating it. So these are just, different scenarios how the macrophage and the B lymphocyte act as antigen presenting cells to the helper T cell. So not only does the helper T cell need an antigen presenting cell to present the antigen to it, it also needs some cytokines to be activated. So for example, interleukin-1 and tumor necrosis factor that are released by the antigen presenting cells are going to help in the activation of the helper T cell. So what about the cytotoxic T cell? Cytotoxic T cells are mainly reserved for identification of cells that are either infected by a virus or are becoming cancerous cells. And that is why cytotoxic T cells don't interact with CD4s, they interact with CD8s. They don't interact with MHC2s, they interact with MHC1s. Remember that the MHC1 are found on every single cell in our body with the exception of RBCs. And all of our cells might be exposed to either changing into a cancer or becoming infected by a virus. So the cytotoxic T cell interacts with that class 1 MHC. If again, it is able to identify it as a normal class 1 MHC, that means it's a self cell, it is not going to attack it. But let us say in this scenario, you see this cell is infected with a virus. So part of the virus is going to be presented here on the class 1 MHC, so making it an abnormal in shape. That cytotoxic T cell recognize it that it is not normal and is going to start attacking that cell. Now, how does that cytotoxic T cell attack it? Well, remember the cytotoxic T cell has to be activated by the helper T cell. So different cytokines are going to activate that cytotoxic T cell. Now that cytotoxic T cell is going to make a hormone or a paracrine known as perforin. Perforin literally does what the name indicates. It is going to make different holes or perforations in these infected cells, basically killing those cells. Natural killer cells, those are a kind of lymphocytes that are very similar to cytotoxic T cells. Actually, they were thought to be a third kind of T lymphocytes, but now they are considered a different category of lymphocytes. And their major targets are also cells that are infected with viruses or cancerous cells. These natural killer cells have the ability to attach directly to the cells and killing them. They perform what is known as a surveillance, where they pretty much just survey the cells for any cancerous changes and kill them right away. Now, how do these activated helper cells activate macrophages and natural killer cells? That is through the release of interleukin-2 and Interferon-Gamma. So you want to know these two cytokines, interleukin-2 and Interferon-Gamma, that are released by the activated helper T cell in order to further activate macrophages and natural killer cells to eventually destroy their target cells. Now, how does our immune system, how do our immune cells recognize our self cells? This is a process that occurs during fetal development and very early in postnatal life. It happens, it's a two-step process. The first step is known as clonal deletion or central tolerance. And this happens in the primary lymphoid tissue. So it happens in the thymus and in the bone marrow. And what happens is that the T and B lymphocytes are going to be exposed to all of our self-antigens for the first time. So they're pretty much introducing our immune system to our self-antigen. This is what my MHC1 looks like. So whenever you see this, do not attack me. Okay, so the T and B cells that are capable of reacting with these self-antigens are going to self-destruct. That process is known as apoptosis. That again is known as clonal deletion or central tolerance. But when the T and B lymphocytes migrate to the secondary lymphoid tissue, so the lymph nodes and spleen, this is where the second step happens, and that is clonal inactivation or peripheral tolerance, where again, if we find B and T cells that have the potential to react to our self cells, they are going to become deactivated. Obviously, this is not a perfect process because we still developed autoimmune diseases. And an autoimmune disease is the failure of central and peripheral tolerance from developing. Now our B and T cells look at our class 1 MHCs and think of them as invaders and start attacking these cells. And that ends this portion.